I saw your email. I'm just trying to figure out my schedule to see what days work, so I'll get back to you. Any chance, Abby, you can possibly take it before, or would it have to be after? Like the exam Thursday, and I know you said you're not gonna be here Thursday and Friday, so if you took it on Wednesday, is that possible? Is it? Okay. Just so I'll confirm, before then with you, but just to know if that's ours.
Who's the real winner here?
So like what also did you add that so I can do the next one? So I write it to be that order. This is like on the website, like this is like a citation.
Last time I think I just.
Then I'll just do this. And then on the bottom, is it just like this or just straight? Like such.
This building can kind of be like the science open version.
Late summer issue.
All right, everyone, good morning. How's everyone's weekend? Yeah. All right, well, this is the last week of class. So we left off lecture 19, the very end last week on Wednesday. So I'm just gonna pick up from there, and then I'll pull up lecture 20 on the transmission of infections, and we'll just go on from there. At the end, before you leave, pick up a worksheet number 8. This will be the last worksheet you'll have. So you will, use this in worksheet 7 as like a study guide for the exam. And so I'll talk a little bit about that more later, but just make sure you pick up a worksheet before you leave. So I guess I'll start by asking, is there any questions at all about anything you guys want me to go over from last week before I jump into this here, this figure here? Okay, so we were talking about different places that a microbe can infect within a cell or, outside of a cell. And so the two places within a cell is in the cytosol, as we talked about, and then you can have, here are the pathogens in red, and then also you can have the pathogen inside of the cell, but also inside of a vesicle in the cell. And then pathogens can also be extracellular, so those would be ones on the outside of the cell. So for immune cells that are recognizing antigens that are coming from an MHC class 1 molecule, so what that means is we have immune cells in our body that have these MHC classes that are essentially just these like proteins on the cell, and these are what are binding to the particular antigen and then presenting it to our T cells or B cells, the adaptive immune cells. And so there are certain cells that, all cells actually can have this MHC class 1. And so those cells will have to present the antigen peptides on its surface to T cells. And so it can do this in two ways. And so these MHC class 1 molecules usually are on, an infected cell, and they can directly present it to T cells, or it could be more indirect, where we call that cross-presentation. And so that's when we have these antigen-presenting cells, so like dendritic cells. They take up the antigen from other infected cells and then present the antigen to the T cells. So this is direct, this is more indirect, the cross-presentation. And so there's this other concept called cross-priming, basically activating other naive T cells via this cross-presentation process. But it's important to know the two types, direct and cross-presentation of antigens, and this is only relevant to cells that have MHC class 1 molecules on their surface. For antigens that come from cells that have MHC class 2 molecules, then that is going to require antigen-presenting cells that will capture the antigen and then present them on the cell surface. And the cells that do this are macrophages, dendritic cells, and also B cells. So if you remember, macrophages and dendritic cells are the two types of antigen-presenting cells that we talked about. And so macrophages, they engulf via phagocytosis. Dendritic cells are activated effector T cells. And then B cells are adaptive immune cells, and so they can also capture antigens by a B cell receptor and then present it to T cells. So so it's important to remember where the antigens come from if you're looking at an antigen that's attached to an immune cell that has MHC class 2 molecule versus MHC class 1. So kind of to put this all together a little bit, MHC class 1, usually for the most part, not always, but we're going to just kind of go with this grand paradigm that it's intracellular antigens that are being recognized by cells that have MHC class 1. So foreign proteins, say, a virus or something, that it's in the cytoplasm, and it's, this is a virus protein, and it's starting to be degraded into these peptides. So these peptides are the antigens from the microbe or the virus. And so here we have these immune cells that have, this one has an MHC class 1 and 2 on it, but because this is a virus and it's being made in the cytoplasm, it's going to be recognized by MHC class 1 molecules because it's intracellular. So they are loaded onto this MHC class 1 on the cell, and then it's moved through different complexes in order to get it out of the cell and present it to something else. So just showing you this, if we have an antigen that's extracellular, generally that's going to be fused or taken up by a MHC class 2 molecule on the cell, and so that's just kind of the key thing here is to remember that MHC class 2 molecules more likely will recognize extracellular antigens, and MHC class 1 molecules are the ones that pull in the intracellular antigens and present it to the T cells. You don't have to know these little nitty-gritty details, but this main part is what's highlighted. Just another quick, image with more information than you need to know, but just kind of showing you, really wholly encompassed image of everything that can go on in these primary and secondary immune responses. And so when you have an intracellular pathogen, we're talking about these MHC class 1 molecules that are binding. And so remember, MHC class 1 molecules are recognized by CD8 protein receptors. And so this will, if you go back a few slides, we talked about how CD4 is what's going to be recognizing the MHC class 2, and CD8 is recognized by the MHC class 1. It's the way that I remember it is kind of difficult to get that straight, because you say so CD8, we have CD4 molecules, and so MHC class 1 recognizes CD8, MHC class 2 recognizes CD4. I don't know, the way that I remember it is you have 1 and 2 and 8 and 4. 1 comes before 2, but when it comes to the CD receptors, it's not necessarily the smaller one goes with the smaller one, it's the opposite. So 1 goes with 8 and 2 goes with 4. It doesn't really make any sense, but I try to make sense of it that way. It's not intuitive. It's not like 1 is going to match with 4 and 2 is going to match with 8, because those are the larger ones. It's just the opposite. So that's how I remember it. You can use that, or it might confuse you more. So, but you should remember that. And so when we're talking about extracellular pathogens, usually humoral immunity is favored, and so humoral immunity involves B cells, and B cells really are, basically are the B cells that have the MHC class 2 molecules. So they are going to be activating other T cells by binding to that CD8 or CD4 molecule. And so one thing that our immune systems do that's really really efficient in our lifetime is this persistent protection during secondary responses. So our body continues to create and secrete antibodies. So plasma cells, which are a form of mature B cells, they will continue to secrete antibodies for several months even after an infection ends, and so they help to confer this protective immunity. Plasma cells are in the bone marrow, because that's where B cells are made and being matured, and then they are also in the tissue beneath mucosal surfaces, and so even after months have passed after an infection, this immunity will continue to be secreted through these antibodies. Eventually, this immunity will go away, but subsequent exposure to the pathogen will result in a secondary immune response. So here, I'm showing you a graph where over time, we're looking at the type of immune response. So the first time you see an infection, you don't have any immune response to it at all. And so over time, your body will start to, generate these antibodies. So those are B cells, and then also affect your T cells. So this is your adaptive immune response. And then over time, it's, even though like after a few weeks, you probably don't have any infection anymore, you're still generating those types of antibodies. You go several months to years, and it kind of stays low. You have this protected immunity or immunological memory. And then if you have a secondary infection, or if you experience that pathogen one more time, your immune response shoots up higher than where it even began the first time, because of this memory that your body has, this protective immune response through these memory B and T cells. So you get this high secondary adaptive immune response. So these long-lived memory lymphocytes, so those are both, the plasma cells and these T cells. So after about a year post-infection the circulating antibody decreases, but a steady level is maintained for life. So this is kind of the point here where this is still a really low level of it. It's not completely gone as to the same way it was during the first exposure to the infection. So these are long-lived plasma cells. And so these cells and their secreted antibodies become part of immunological memory. They're high affinity antibodies. They're greater in number than naive lymphocytes because of that concept of clonal expansion that we talked about, where they continue to be regenerated. And so they replenish in the bone marrow by proliferating. Any questions? Not, I will move on to lecture 20. We're going to kind of go away a little bit from this concept of like immune system stuff. We're going to kind of go back a little bit to infections, and actually like zooming back out to the human and the host and how infections can be transmitted. And so just a reminder, the final exam is on Thursday, May 2nd. It's going to be at 10:20. So just keep that in mind, that it's not going to be during any of the class times for this class. We were scheduled for it to be on Thursday. I personally will not be able to be here for the final exam. I have a conflict, but one of the other professors will be here to proctor. So, please take advantage of Wednesday in asking your questions, and we'll review, all the material for this exam. It won't be cumulative. It will only contain, content from Unit 4. So I believe that was like lectures, 17 through, probably more than that, like 15 or 16 through 20. But yeah, so let me know, any questions you have. I'll also be available via email, but I won't be here during the actual final exam. And if you do have a conflict with this, let me know so we can schedule something in advance. So we're going to kind of go a little bit back to this whole concept of the different mechanisms that pathogens have to cause infection and ultimately cause infectious disease. And so, they can cause infection by just getting into the body and adhering, and then acquiring the nutrients they need, being able to persist and multiply, avoiding the innate and adaptive immune responses, and ultimately causing some tissue damage. That's the infection. And then once that pathogen is able to exit from the host and transmit the infection to a new host, that's when we call it an infectious disease, because now it's being transmitted. And so microorganisms can be transmitted from host to host, or even acquired by a host, by multiple mechanisms. The two, overarching themes are horizontal transmission and vertical transmission. Horizontal transmission is transmitting a microorganism from one individual to the next, so individual to individual. Vertical transmission is transmitting from the maternal to the offspring, so maternal to neonatal. So the infectious agent can cross the placenta from the mother to the child, for instance. So for the rest of this talk, we're going to be focusing more on horizontal transmission of microorganisms, but it's important to know that vertical transmission is also, a really important one to note, and so that's why, pregnant mothers have a lot of restrictions so that, they don't get an infection and pass it on to the offspring while their immune systems are still developing and pretty immature. So obviously one of the main contacts or mechanisms of transmission horizontally is direct contact from person to person. So that's usually through the spread of microorganisms through actual physical contact. So it's not sneezing and somebody getting it through the air, it's sneezing on your hand and then shaking your hand with somebody. So that would be direct contact transmission. Indirect contact transmission occurs when a susceptible person comes in contact with a contaminated object or contaminated material like soil, water, etc. So that could be, like you touching a doorknob after somebody just coughed and, put their hand on the doorknob. And so in healthcare settings, virtually any item could be contaminated with certain microorganisms. That's what leads to those nosocomial or hospital-acquired infections. And fomites are inanimate objects that can be capable of transmitting the infectious organism from one individual to another. So if you think about this in the hospital setting, that could be doorknobs, beds, sheets, anything like that, furniture, clothing. So anything that's inanimate can carry, bacterial infection, and when a susceptible person comes in contact with it, then it can be transmitted through them indirectly. So we're only talking about inanimate objects here. And so this one is a little bit, easier to spread bacteria because bacteria don't require a living host, but a virus cannot really be transmitted through this because a virus can't survive on a non-living host. So this one would be specific to bacterial infections. Any questions so far? Okay. So human-to-human disease transmission and trying to control them. So fecal to oral spread, that's obviously one of the main ways that we get, a lot of different bacterial infections. And that's, what we were doing in lab last week where we were trying to, see if there was some type of fecal-oral contamination in the water experiment. And so this is generally controllable by public health measures. Another human-to-human disease transmission mechanism is venereal spread. So that's basically, having sexual contact with people, and that is way more difficult to control because social factors are involved. It's also really important, and it's difficult to control because a lot of times these, diseases that somebody can have are asymptomatic or they can be latent. So it can be a virus, for instance, that somebody has, like HSV or herpes simplex virus. That is one of the symptoms of that is having cold sores, but that can also, be something spread through your genitals. And so that's something to really consider because even if you don't have the cold sores, doesn't mean that the virus isn't there. So it's important to know your status just for any STIs or STDs because it's really hard to control. There's so many social factors involved where people just don't get tested or just don't know enough. And then lastly, respiratory or salivary spread. That's also not readily controllable. Mainly because, also especially in like college towns, people are exchanging saliva in various ways. But beyond that, these types of respiratory infections are spread through aerosols or spread through, little tiny droplets in the air that you can't even see. So that really, causes the infection to spread and it's hard to control. So just important little reminders: wash your hands, know your status, and wear a mask to kind of help control these different types of infections. So another mechanism of horizontal transmission is inhalation. So that usually will lead to infection via the respiratory tract. You'll inhale something and then it'll get into your respiratory tract, and that's usually through these aerosols. So aerosol transmission is the mechanism. And so sneezing and coughing without covering your mouth spreads droplets, that's these aerosols, that could be infectious. So here, this is a video of a high-speed camera that captures how different types of face masks work. Using different types of masks. You can literally see like the aerosol spread after a cough 10 times. With no mask, you see the aerosol spreading. You have a cloth mask, you have a lot less, but you still have some. Same thing with this mask. And then if you wear a surgical mask, there's like virtually no aerosols that are being able to be spread. So it's just, you know, reiterating the fact that these aerosols and wearing a mask in general really does protect you tremendously. So it's important to cover your mouth when you are you know, coughing or sneezing. Just another, you know, little, you know, experiment or, you know, paper that came out that is showing this, this contribution of masks and how they control airborne and aerosol transmission of infections. So, you know, even speaking, infectious aerosol particles can be released during breathing and speaking by asymptomatic individuals that are infected. So not masking will maximize your exposure, and having universal masking results in the least exposure. And so that's why, you know, with coronavirus, masking became, you know, one of the things you had to do. And just kind of showing, you know, the different types of having an infected individual not wearing anything, and then an uninfected individual, and like how, you know, what is this, the analysis, so this is the delay of transmission. So things will be delayed a lot more if you're wearing more masks, wearing a better, more protective mask. So there are other factors that affect indoor airborne transmission beyond, you know, you just, you know, covering your mouth when you're sneezing. So droplets can be different sizes. So here you can see like particle size. They could be tiny or they could be larger. And so usually the motion of large droplets is governed by gravity, and they will, you know, just fall towards the ground because that's the law of gravity. But the movement of aerosols, these really tiny particles, is more strongly influenced by airflow and the direction and pattern of that airflow, as well as the type of ventilation and the air filtration and disinfection. So you can, you know, be sick in your home and just coughing all over the place, and, you know, you might get better, but if you're not properly ventilating and airing out that space, then you're going to have these aerosols that are moving around and strongly influenced by the airflow. And so, you know, it's important to have, you know, proper ventilation, you know, opening up windows and letting some natural air coming in and out, and then also air filtration. I have an air purifier in like every room in my house just because it just helps me breathe better, fewer infections, fewer allergies. My asthma flare-ups are a lot better. It really does help. There's so many studies that show that that those kinds of things really, really do help. Yeah, so Any questions so far on any of this? Okay. Another way to horizontally transmit infection is through ingestion. So we call this the F diagram because really when you're ingesting something, it's coming from fecal contamination. So you don't wash your hands properly. There's a break in sanitation, so your feces can get into your fluids or on your fingers or flies or fields or floors. So this is the F diagram. And so that, can then get into your food, and then you can contaminate the future victim. Because this is due to fecal oral transmission, infection will be via the gastrointestinal tract. It's coming from the gut and it's being passed on to somebody's gut essentially through these different mechanisms like fluids, fingers, and the others. An example of this is the fecal spread of EHEC, or enterohemorrhagic E. coli. And so, like we've said before, most E. coli strains are harmless, but there are some that are potential pathogens or are pathogenic. And so here, EHEC, the infection source is cattle. So cattle and other ruminants are the main carriers. So it comes from a cow. And then so that can then get, into, or when, we're eating our meat, uncooked meat and raw milk can carry that infection or this E. coli. And so, if you're not heating or cooking it at a high enough temperature, then the bacteria doesn't die. And so, that can then also get into, you eat that uncooked meat, pathogen gets into your body and into the GI tract, and once it's in the human stomach, it starts producing toxins that cause serious illness. So this is one of the toxin-producing E. coli strains, and so some symptoms, because it's an enteric bacteria, it's hitting your GI tract, you're going to get this diarrhea, sometimes bloody diarrhea if it starts to rupture your internal organs, fever, vomiting, muscle spasms. It can get really, complicated in immunocompromised individuals and cause hemolytic uremic syndrome, so blood in your urine. The death rate is 3 to 5%. So it's one of those, it's not, obviously 3 to 5% is still too much, but it's not as virulent, I guess, as what we talked about previously with Listeria, that intracellular pathogen. Bringing that up again, because you guys should know that one. So giardiasis is another infectious disease, and so this is the last one we'll talk about. This is caused by infection with Giardia lamblia. So what this is it's essentially a parasite, and it can get into the intestinal tract of many different vertebrates, so not just humans. It can get into dogs, beavers, sheep. So that it can then be transmitted between different hosts. And so the transmission of the disease is primarily through the ingestion of water or food contaminated with these cysts. So it forms these cysts, and then when it's usually out like in the soil and stuff, and so these can then get onto veggies when they're harvested, and then you buy it, you don't wash it properly, the cysts get into your body when you eat it. And even if you ingest just a few amount of cysts, so in this case a few is about 10, that's considered infectious because these cysts can survive for months, even in the cold, and so the disease can spread very, very easily, and it can occur in daycare centers. So that's one of the ways that this can get around in daycare centers with kids. So, there's many different routes of transmission. Through the oropharynx, so your saliva, respiratory tract, coughing, sneezing, gastrointestinal, fecal, your genital tract through your urine, STDs, perinatal, skin, milk, and blood. Blood, if you can get infected through needles and bite marks, and so those are all routes of transmission. but it's going to be transmitted to something else. So that something else is going to have to acquire the infection. So we call that a route of acquisition. So you need to have both. So basically you can acquire it through your respiratory tract, your mucosal surfaces. You can inhale it through your nose or in your mouth. GI tract, same thing, your genital tract, conjunctiva or the eyes. You can even have infections get in through the eye. And so the important thing here is that the route of transmission has to match up with the route of acquisition, as well as the infectious agent's mechanism of infection in order for disease to occur. So it seems like it's got to have a lot of different, check marks in place, and there are different factors that affect, whether disease will occur. So it's just important to know that, if I'm coughing and sneezing and I'm transmitting something through my respiratory tract, it's not going to cause infection unless there's a recipient that's going to acquire it through either their mucosal surface or their respiratory tract. So it's got to match up in that way. And then the other thing to make sure, to note is that the infectious agent, the mechanism of infection. So if, I'm coughing and sneezing and you're, around me, but that type of bacteria, only, I don't know, I guess will stick onto some food source and you haven't eaten that food source, then you're not going to get infected because the mechanism of infection has to match up to the route of acquisition as well as transmission. There's so many variables that will, affect this, this ability to infect, and on top of that, there is this dose of infectious inoculum. It's a really critically important variable. So how much you're getting. So the dose. So I guess I just gave an example of, having as few as 10 cysts of the Giardia lamblia can cause this, really bad infection. So that would be a dose. So whether it's cysts or number of aerosols you're inhaling, or just number of cells that you're getting from, from somebody, that's going to also impact the disease and whether it occurs. So lastly, I just want to kind of talk a little bit about this variable in epidemiology and infection control, this RO. This is called the reproductive number. And so what this is the average number of people who will contract a transmissible infection from one person with the disease. So it's the average number. So if that number is 1, it means that someone with the disease will pass it to one person. If the number is 2, then each infected person will pass it on to 2 people, and then those 2 people will each give it to 2 more people, and so on. So the greater the number, the more infectious it is. So if the number is greater than 1, then obviously the number of people with the virus will rise, and if it's less than 1, then the number will decline. So public health officials are always trying to control infectious rates and bring that reproductive number down. There's a link here to just a little website, scientist.com, where it talks about why RO is problematic for predicting COVID-19 spread. If you're interested, it's just like a little FYI. But the RO for SARS-CoV-2 infection is estimated to be around 4 to 7, so it can still be a little ambiguous, but it is pretty infectious. Just to give you a little bit of context of other infectious diseases, where smallpox has an RO of 3, and then measles has an RO of 16 to 18. So one person can infect 16 to 18 people, and those 16 to 18 people can each infect 16 to 18 more people. So it really, shoots up on the scale. But just kind of keeping in mind, these RO values and what they indicate. If I were to give you a chart of different RO values and were to ask you which ones were more infectious or not, you should be able to tell me that. Any questions? Okay, so that's all I have for you. I I know that the previous couple of lectures were a little bit more in depth on immunity, so I have a worksheet that you guys should complete before Wednesday. We'll go over it on Wednesday. We'll go through all of the lectures and any questions you have for the final exam, because that will be the last time we meet. And if you did want to meet, you know, for like a private appointment or office hour, then I can definitely do that as well. So just email me and let me know.